let's look at one or two more issues to do with the formation of protein before we finish. I mentioned amino acid charts. Here are two examples. This one's circular. It has the names on the outside of the 20 amino acids. And here we have the code divided into codons. Remember, 3 bases, say guanine, guanine, uracil, or guanine, adenine, guanine here. Each of these is a codon, so 3 of those are, another word for it is a base triplet. These are examples of, or I should say another way of describing a codon. So each of these provides the code for one amino acid, and this is read by the transfer RNA, tRNA, in the ribosome. So if a sequence of RNA and messenger RNA arrives at the ribosome and it has a certain part of the sequence, then let's see if we can identify what amino acid this goes for. Here's an example of a sequence that begins like this. If you look here, GUU on the RNA represents this one here, which is called valine, V-A-L-I-N-E. That's one of the amino acids, so that one will go for valine. And then the next section, when it's red, I think I may have moved back to front, but it doesn't matter, get the idea. CUU, which is here, is leucine, L-E-U-C-I-N-E. While you're not expected to know too much information about amino acids, I think it's very valuable to know that a code relates directly to the protein by the choice of amino acids each one codes for. I'm now going to add some keywords related to this. We've just seen an example of a codon or a base triplet, which provides the code for one amino acid. mRNA is the conversion from DNA to RNA so that it can leave the nucleus and go into the cytoplasm. But tRNA is in the ribosome, and it is the one that reads that codon. So what is a gene? We have to look at it from two different directions. From the bottom, we can say it's a section of DNA with thousands of bases. This then becomes a protein. This is if the code is expressed. But another topic of biology which we haven't done yet is gene expression. We all inherit two different sets of genes from both parents. And in this case, we're just talking about when the gene is expressed, when something happens. So you can also say that a gene is part of a chromosome. So a chromosome is made up of genes, and those genes are made up of DNA. So we're talking from macrostructure, as in big, down to microstructure, as in smallest. So to finish, at a molecular level, we mean DNA or RNA, so that's a biomolecule. A large section of DNA then is a gene because it does something when it's expressed. By the way, I think the coronavirus has about 10 genes, so it's very efficient. It doesn't need to do much. And then genes are lined up along a sequence to form a chromosome. So the chromosome is the close structure. And all of the chromosomes, humans have 23 pairs, are packed tightly into the nucleus, supercoiled around each other. And when they're not replicating, they form a mass called chromatin. So the chromosomes are not identifiable. They're just in a mass called chromatin. Later, we look at individual chromosomes when we're talking about genetic profiling.